Hello everyone and welcome to ITLS Academy Empower the Youth. So today in this lecture we will going to study about density gradient centrifugation. So in the previous lecture we have discussed about what is centrifugation. We have seen the different types of centrifugation. We have discussed about ultracentrifugation and its principle of working, that on which principle ultracentrifugation basically work and what are its part that is involved in ultracentrifugation device, whereas we have seen its application and on the basis of its principle, its working we have discussed. So in this lecture we will going to study about density gradient centrifugation. So before understanding its definition, let us understand the introduction part of this. So the introduction part says that there are two types of centrifugal techniques for the separation of particles. So we all are familiar with this term that the centrifugation is basically used to to separate two different molecules, or you can say particles. It is also used in the purification technique, but it is largely and basically used in the separation of particles, whether the particles or the is of same, like two liquid or sometimes when it is two solid. So basically its main work is of separation of particles. So the introduction part says that there are two types of centrifugal techniques for separating the particles. So what are the two different types of centrifugal techniques are there? The first one is differential centrifugation and the second is density gradient centrifugation. So the first one is the density gradient differential centrifugation, whereas the second one is the density gradient. So the density gradient can further be classified into other parts like zonal, rate zonal and isochronic centrifugation. So it has a further distribution, or you can say it has divided into the two parts, that is the rate zonal or you can say the isochronic. So what is basically rate zonal and the isochronic? So we will going to discuss in this particular lecture. So the differential centrifugation, rate zonal centrifugation and isochronic centrifugation, and the last one is the suitable density gradient medium selection. So what is density gradient centrifugation? So centrifugation allows researcher to separate cell based on their shape and size. So we have already discussed this term that the researchers, they use centrifugation techniques to basically to separate a cell on the basis of what? On which basis they separate? On the basis of size and shape. Samples are placed into centrifuge and further the machine is designed to spin liquid solution at a set speed. So the samples, what they do, they are placed in the centrifuge machine, and on the basis of the shape and size they are separated with the help of a centrifugal force, and the machine is designed so that they spin the liquid solution at a particular speed. Means there is a set of speed that we have already discussed in our previous lecture, so you can go to that lecture and see what is the speed, that is 60 rpm to 1,50,000 rpm. So the rotation causes the mixture to experience a centrifugal force that pushes larger particles from the center towards the bottom and smaller ones to the top. The larger component experiences a strong centrifugal pull than the smaller component. So since the rotation, what does this rotation do? They causes the mixture they causes the mixture to experience the centrifugal force, and sorry, because of this centrifugal force, or you can say due to the centrifugal force, the particles that are larger in size, they are placed at the bottom, and they are you can say they are moved towards the bottom because of its size, and the particles that are basically of smaller size, they are passed to the pass through the upper part of the uh, you can say at the top. The process is similar to the density gradient centrifugation, where the samples are also placed into the centrifuge, but the end goal is not to sort them by the size. Spinning from the centrifuge causes more dense particles to move to the bottom of the tube, because these particles have mass, more mass are carried further by their inertia. Less dense particles then settle higher within the sample. This process creates a sorted solution layered by the particle density from least to most, or top to bottom. So here it is not the way that you are prioritizing only the or you can say you are doing on the basis of their size. No, it is not so. The particles are sorted on the basis of the shape, size and density, as well as on the basis of their masses. So this is the criteria behind the separation of particles by centrifugation, that is density gradient centrifugation. So here it is making them making easy for a particle to get separated at a particular speed. Now comes to the principle of the density gradient centrifugation. So in the previous lectures also we have discussed about the principle. So as already told you that these principles are based on the sedimentation principle, means it is same as of the sedimentation principle. So let us discuss this its principle in detail, and let's understand what does the principle mean. So if you if you see their principle, then each particle has a specific set of physical characteristic, the properties of its biological makeup that can be used for the separation and isolation. Then comes the density gradient centrifugation, focuses on two characteristics. The first is size and second is density. So this term is very important, that on which criteria, means on which type it is focusing. So basically the density gradient focus on two factors, or you can say two characteristics in any of the sample, or in any of the particle. The first one is shape, and the second one is its size. Or you can know, sorry, the first one is the size and the second one is its density. So on this basis, or on this criteria or characteristic, the density gradient generally works. So the length of the time required for this procedure depends upon the particle size. Larger particle will reach their position of stability earlier, while the smaller particle take longer to pass through the larger particle zone and assume a position deeper in the gradient. Now comes the density gradient reagent. So what are the reagent of density gradients, or what are the reagent that are used in density gradients? Now let us understand this. In density gradient centrifugation, distinct reagent with specific densities are used to isolate or separate the cell. So, depending upon the shape and size and density, different types of reagents are used for the isolation of the isolation or for the separation of the cells. So not only can these products speed up the processes, but they can also increase the purity through and throughput. By keeping the particle from clustering and creating a distinct interference, the quality of the reagent can greatly increase the efficiency of density gradient centrifugation. Centrifuge component. So the centrifuge components, the centrifuge spinning mechanism is composed of two distinct parts: the rotor assembly and the electric motor. The electric motor provides the rotation power, and rotor assembly transmit the rotation energy into the rotor tube. The rotor assembly will hold the density gradient tube, which are prepped with media of known density to aid in the layer separation. So if you talk about the reagent, so reagents are used for the isolation as well as for the separation of particles. So apart from isolation and separation of particles, these reagents, or you can say these reagents are also involved in the purification of particles. Apart from purifications of particles, they are also involved in other things, that is, in the purity of the particles, means they are increasing the purity of the particles, or you can say result and second is the throughput. Now comes the centrifuge component. So basically here two distinct part, the centrifuge spinning mechanism, it is, they are composed of two parts, distinct part. The first is the rotor assembly and second is the electric motor. So in the interviews also this question may arise, that this rotor that the centrifuge spinning mechanism is depend on which two or factors, or on which distinct parts. So first is the assembly motor and the second one is the electric motor, right? Electric motor, and second one is your assembly, rotor assembly. So this questions may generally arises in the interviews or in various type of examination. So you must be aware of these two component. Now let's quickly discuss about the application of density gradient centrifugation. So as far as we have discussed different types of centrifugation and techniques, so we have generally seen that all these techniques are involved in the separation of particles. They have a great applications in different types of biology lab, and in also they are involved in the purity of different types of samples. So they have a vast application in different types of industries, like in the examination or in the separation of proteins, RNA, DNA. So they have a vast application. Now every type of gradient has its unique feature that is involved in different types of industries and different types of labs. So let us discuss the applications of density gradient centrifuge in detail, and let us understand what are their application and how they are useful in different types of factors. So the gradient centrifugation is used to purify sample into their component population. While the cell separation is a popular application of density gradient centrifugation, it can also be used to determine the unknown densities of different particles. Centrifugation of all types of benefit researcher because it harvests substance for the additional experimentation or medical use. It helps to improve dead cell and debris in sample so that the specific groups of cells or particle may be effectively isolated and studied. So it has a great application in isolation, and it also helps or you can say benefit the researcher in many ways, like for and for like for example, it helps to remove the dead cells and debris in the sample. And they have apart from the experimental approach, or apart from the experiment application, it also have a medical application as well. And it is widely studied by or helping, or you can say giving benefit to different types of researcher as well as scientist. Now comes the differential centrifugation. So what is differential centrifugation? The simplest form of separation of centrifugation is differential centrifugation, sometimes also called as differential pelleting. Particles of different densities or sizes in a suspension will sediment at different rate, with the larger and the denser particles are sedimenting faster. These sedimentation rates can be increased by using a centrifugal force. A suspension of cells subjected to a series of increasing centrifugal force cycle will yield a series of pellet containing cell of decreasing sedimentation rate. For example, you can see this diagram. The particles that are lighter in the weight, they are collected at the bottom of the test tube. Particles of different densities or size will sediment at different rate, with the longest and the most dense particle sediment faster, followed by the less dense particle, as you can see this image. If you see this image, like the image 1, they have different varieties of particles. Some are very small, some are in the medium size, some are larger in size. So if you see the green one are on the are very large, so they settle at the bottom first because of its larger size, then followed by the red one. They are have a less medium, or you can say medium size, so they settle second. Then the blue one, they are shorter than the red one but bigger than the black one particle, so they settled before the black one. And at the last, because of its size, because of its density, so they settle down at the bottom, means later. So the particles of different densities or size will sediment at different rate, with the largest and the most dense particle sedimenting the fastest, followed by the less dense and smaller particles. Differential pelleting is commonly used for the harvesting cell or producing crude subcellular fractions from the tissue homogenate. For example, a rat liver homogenate containing nuclei, mitochondria, lysosomes, and membrane cell vesicles that is centrifuged at low speed for the short time will pellet mainly the larger or more dense nuclei. Subsequent centrifugation at a higher centrifugal force will pellet particles of the next level, or you can next lower order of the size, and so on. It is unusual to use more than 4 differential centrifugation cycles for a normal tissue homogenate due to the heterogeneity in the biological particle. Differentiating centrifugation suffer from contamination and poor recoveries. Contamination by different particles type can be addressed by resumptions and repeating the centrifugation step. Rate zonal centrifugation. So, in the rate zonal centrifugation, the problem of cross contamination of particles of different sedimentation rate may be avoided by layering the sample as, as a narrow zone on the top density gradient. So, in this way, the faster sedimenting particles are not contaminated by the slower particles as occur in differential centrifugation. However, narrow load zone limit volume of sample that can accumulate on the density gradient. The gradient stabilize the band and provide a medium for increasing density and viscosity. Sample is laid at a normal zone on the top of the density gradient, that is 2b. Under the centrifugal force, particles move at different rate depending upon their masses. The particle which have mass more, that will come at the center very fast as compared to the particle which has a less densities or mass will occur later. The speed at which the particles sediment depends mainly on their size, mass instead of density. As the particles in the band moves down through the density medium zone, containing particles of smaller size form as faster sedimenting particles move ahead of the slower one. Because the density of the particle is greater than the density of the gradient, all particles will eventually because the density of the particle is greater than the density of the gradient, all particles will eventually form a palette if centrifuged through a long enough. So this is somewhat a diagram look. So before centrifugation it is look like the sample zone. This is a sample zone and it has a very it is in a low density medium, and then after centrifugation the larger one settle down, medium will go in the in the middle, and the smaller or particle which has a low density will come up or come on the top. Now, what is the isochronic centrifugation? So, isochronic centrifugation, in isochronic centrifugation, also called as Bionent or equilibrium separation, particles are separated solely on the basis of their density. Particle size only affect the rate at which the particles move until their density is same as the surrounding gradient medium. The density of the gradient medium must be greater than the density of the particles to be separated. By this method, the particle will sediment to the bottom of the tube no matter how long the centrifugation time is. Then comes the starting with a uniform mixture of the sample and the density gradient, and under the centrifugal force particles move until their density is same as throughout the medium. This is again very important. Upon the centrifugation of particle of specific density sediment until they reach the point where the density is same as the gradient media. Gradient media means the equilibrium position. The gradient is said to be isochronic and particles are separated on according to their buoyancy. Since the density of the biological particles is very sensitive, So because of its sensitive to the osmotic pressure of the gradient, so isochronic separation may be very significantly depending upon the density gradient used medium is used. Although the continuous gradient may be more suited for analytical purpose, preparative techniques commonly use a discontinuous gradient in which the particle band at the interference between the density gradient layer. This makes the harvesting certain biological particle, example lymphocyte, easier. In this we understand that the lymphocyte sites cells are they get separated by this technique. Now comes the suitable density gradient and medium selection. So means on which basis it is selected. So the primary function of the density gradient centrifugation and is to separate the particles, that we have already discussed. Means what is the basic aim? Basic aim or basic agenda is to separate the particle either on the basis of their buoyancy or either on the basis of their density, and at last on the basis of the rate of sedimentation. For the isochronic separation, the important feature is that the maximum density of the gradient medium is higher than that of the particle, means on the basis of their buoyancy. So what is an ideal density gradient media properties have? So the first one is the sufficient solubility to produce a large density is required. This is the ideal you can say property. The second is does not form solution of high viscosity in the desired density range. Third is not hyperosmotic or hypoosmotic when the particle is to be separated are osmotically sensitive. Solution of the gradient should be adjustable to the pH and the ionic strength are compatible with the particle being separated. Fifth point says does not affect the biological activity of the sample. Sixth one is a non-toxic and they are not metabolized by the cell. Seventh point says does not interfere with assay procedure or react with the centrifuge tube. Eighth point says that it should have a property that can be used to measure the concentration. Ninth point says they should be very easy from the purified product. 10th says autoclavable, and last is the reasonable cost. So these are certain properties for an ideal density gradient media. Means a media should have all these qualities inbuilt in themselves so that it make them suitable for the use. Now comes what is the difference between the rate zonal and isochronic centrifugation. If you see the rate zonal, the gradient is shallow, whereas here is a gradient is steep. Gradient density is maximum less and than that of the least dense sedimenting species. Same is the speed is of the maximum. Then centrifugation is carried out at a low speed for a shorter period of time, whereas centrifugation is carried out at a high speed for a longer period of time. And the last is this method is used for the separating particle which differ in size, not on density, that is RNA DNA hybrid ribosomal subunit, whereas this method is used for separating particles which differ in density, that is DNA, plasma, lysosome, mitochondria, peroxime, plasma. So this is again the difference. So you can take a screenshot of this thing. I am just zooming in, so you can take the screenshot. Means if you see the definition particle nature, so So in this you check the definition, that is the rate zonal centrifugation. It is an analytical techniques we can use to separate particles of the solution depending on the size but not on the density. But if you see the isochronic, so here the particle is being separated on the basis of their density, not on the size. So this is basically the definition. And second is the particle nature. So the particle differ in size whereas the particle differ in density. In the rate zonal you see that they are time dependent, they are independent of time. So the gradient nature is here the flat and here it is of the steep. Palette formation, the palette are formed. So in this isochronic no palette is formed. If you talk about the application, for the separation of cellular organelle and protein it is used, and for the separation of nucleic acid this is used. So that brings to the end of the lecture. I hope you have understood the lecture. If you have any doubt, any queries, you can simply leave your queries in the comment section below. So thank you students for watching. Please don't forget to like, share and subscribe. And you can also visit our website, that is www.itlsacademy.com. Again, thank you for watching. And yes student, this is our website, that is www.itlsacademy.com. And this is our helpline number, that is 7080833450. You can simply call on this number and get updated of related to any courses. Thank you.